oncology area of research here at the Vall d'Hebron Research Institute, we have several groups addressing the main unsolved issues in cancer research. So our main strength is that we are located in the most important hospital in Spain, and the main strength is, as I said, our translationality, meaning that we work with patients and we correlate our research with the pathology of the oncology department. The molecular pathology group consists with several basic cell and molecular biologists working together with senior medical pathologists of the Department of Pathology. The group is led by Dr. Santiago Ramon y Cajal, who is also the chairman of the Department of Pathology of the Vall d'Hebron Hospital. The main research focus relates with the models of cellular stress in order to identify a mechanism by which tumor cells are more resistant to factors such as hypoxia, lack of nutrients, and chemotherapy. Well, the key projects of the group included, for example, polysomal RNA sequencing of modulated cell lines and tumor samples to identify novel factors translated during stress and associated with increased tumor resistance. This project is led by Dr. Magda Cessé and Dr. Santiago Ramon y Cajal. They are currently analyzing polysomal RNA sequencing data, and in the future is to identify and evaluate novel factors that are regulated under conditions such as hypoxia and inhibition of cap-dependent translation by drugs including mTOR inhibitors. Another important project of the group is the study of regulation of mRNA and protein translation in cancer under stress situation by the translation initiation factor of IF4E. Project led by Alba Martinez, Santiago Ramon y Cajal, and Trun Osen. They identify a mechanism through which phosphorylation of this factor leads to stress resistance in cancer cells. Another project is the role of YB1 in epithelial mesenchymal transition in breast cancer and the implications for tumor progression and therapeutic resistance. This project is led by Barbara Castellana and Santiago Ramon y Cajal. They identify a connection between IL-6, YB1, and EMT in breast cancer. Other emerging areas of the group include the role of HIR3 expression in breast cancer and sarcomas, led by Javier Hernandez, regulation of tumors and malignant progression by gap junction proteins and their prospects as biomarkers and treatment modalities, area directed by Trun Osen. And the group is also participating extensively in many collaborative studies with other groups in the research institute and in the hospital, the clinical services and the hospital departments. The translational research in pediatric cancer laboratory, led by Dr. Josep Sánchez de Toledo, includes several scientists, senior scientists like Dr. Soledad Gallego, Josep Roma, and Miquel Segura. The group interests are focused on search for new molecular therapeutic targets in children with cancer. The research team is especially focused on developmental pathways as Notch and Hedgehog in rhabdomyosarcoma, as well as epigenetic proteins and microRNAs in neuroblastoma. The group collaborates with industry in the development of new therapeutic molecules to be used as antimetastatic drugs. The group is also devoted to molecular diagnosis in children with cancer, and clinical research is also very active. So they are currently involved in more than 15 clinical trials in pediatric oncology. The prognosis of childhood cancer has improved considerably in recent decades, and survival is approximately 75% in Western countries. However, even with the current multimodal therapies, a considerable number of these patients still relapse and eventually die of metastasis formation. Consequently, pediatric oncologists need new approaches to improve the efficacy of anti-cancer therapies. Molecular diagnosis, detection of microdisseminated disease, and the search for new therapeutic strategies would help to improve the results of the current treatment in pediatric cancer. The experimental hematology group, directed by Dr. Francesc Bosch, which focuses on the study of mechanisms of pathogenesis and progression of lymphoid neoplasms, especially chronic lymphocytic leukemia, the most common adult leukemia. The group also studies the ex vivo effects and mechanisms of action of new experimental therapeutic agents. Moreover, they are specialized in the study of immunologic modulation and response to new treatments and myelodysplastic syndromes. During 2012, the group has identified a new function of the protein ZAP70, which is one of the most relevant adverse prognostic factors in CLL. The group has also described the molecular mechanisms behind the differential response of dexamethasone in patients with CLL according to their ZAP70 status. Moreover, they have established an ex vivo system for preclinical drug testing in CLL, mimicking the microenvironment found in the protective and proliferative niches. For the upcoming year, the group is planning to continue developing the lines of research related to the pathogenesis of CLL and to start clinical trials based on the preclinical tests. They are also planning to open a new translational lines of research on primary central nervous system lymphoma. The Animal Models and Cancer Laboratory, or melanoma group, directed by Dr. Juan Ángel Recio. Dr. Recio's interests are focused on skin cancer translational research. His main interest is to translate the results from the lab to patients as quickly as possible. To that end, he has established a close collaboration with Dr. Garcia Patos, Chief of the Dermatology Department, as well as with Dr. Javier Cortés, oncologist in charge of melanoma patients, to learn from firsthand the real challenges of this disease. This alliance, or the melanoma group, is actively contributing to personalize and improve melanoma treatment at the Vall d'Hebron Hospital. Using genetic modified animal models, patient-derived cells, and in vitro experiments, his lab is investigating the molecular basis of melanoma development and progression to improve melanoma patients' outcome. In particular, they are investigating how oncogenes influence tumor metabolism towards the discovery of novel target molecules for therapy. He's also developing novel animal models to study gene-gene and gene-environment interactions and performing preclinical studies using patient-derived xenografts. It's a group of oncology and molecular pathology laboratory led by Dr. Mathilde Leonard, and which includes the following research lines. First of all, the study or discovery, identification and characterization of new genes, indeed, potential oncogenes, tumor suppressor genes, and microRNAs. Specifically, those which could be responsible of molecular mechanisms of bypassing senescence and that lead to cellular immortalization. Second could be translational research by performing large-scale screenings in different types of human tumors at the RNA level, microRNAs and proteins, for proceeding to the molecular characterization and further identification of their mechanisms of action. And finally, the study of the cancer stem cells' specific properties with the medium or long-term purpose of their effective removal in vivo. Importantly, their removal would allow us to eliminate malignant tumors. The research unit in biomedicine and translational oncology, which include several senior investigators, many students and postdocs, and devotes their efforts basically in understanding the molecular basis of endometrial, prostate, and ovarian cancers. One of these groups is led by the senior investigator, Dr. Rosanna Paciucci, who is working with her postdoctoral fellow, Carlos Fernandez, and the group is devoted to the identification of differential changes in the translation regulation system in the aggressive subtype of prostate cancer. Also, in the search of markers of prediction of resistance to therapy in metastatic prostate cancer. Another group in our unit is the laboratory led by Dr. Jordi Petris, which is devoted in understanding the basic mechanisms that regulate healthy and cancer stem cells. Stem cells reside in most tissues in a quiescent state, but rapidly become activated to both repair and regenerate the adjacent tissues. We are studying several genes involved in different aspects of stem cell activation. The lab is also interested in multi-drug resistance to understand failure in some chemotherapy treatments. Another prostate group, which is led by the senior scientists Andreas Doll and includes also the postdoctoral fellow Mireia Olivan and other many students, also in collaboration with the head of the Department of Urology, Dr. Joan Morote. The projects are focused on the isolation and characterization of new markers for early diagnosis of prostate cancer, as well as prognostic markers or predictive to response to chemotherapy. A major recent breakthrough in the group has been the identification of a set of 3 markers which are giving us a precise diagnosis in urine of prostate cancer with a high specificity and high sensitivity. We are in the process of bringing these markers to the market. Another aspect of this research is to characterize genomic and proteomic signatures which could predict metastatic dissemination of prostate cancer. The most long-standing research line in the lab is deals with endometrial cancer and the molecular basis of endometrial cancer. It's a research line led now by Dr. Eva Colas. Those projects include the discovery of new molecular markers in cancer body fluids, mainly uterine aspirates. Actually, concerning this, we already have a diagnostic kit in the market since July last year. Moreover, we are interested in the characterization of the mechanisms of invasion of endometrial cancer. As an emerging field of research also in endometrial cancer, we are searching new microRNAs which could predict the poor response of therapy in aggressive subtypes of this disease, as well as the identification of more appropriate new strategies of therapeutic strategies. We also developed an animal model of endometrial cancer to mimic development and the course and the outcome of this disease. So more recently, the group is also investing a lot of effort in unraveling the molecular basis of ovarian cancer, which is indeed the most devastating disease that we are approaching. We focus on unraveling the mechanism of regional and distant dissemination by analyzing genomic and proteomic signatures either in the surgical specimen or a cytic fluid or in the metastatic implants in the abdomen. We also have identified several microRNAs which can precautiously predict the resistance to carboplatin chemotherapy, which is currently indeed a major setback in the treatment of ovarian cancer. Finally, I would like to add that in the projects dealing with endometrial cancer and ovarian cancer, we are collaborating very closely with a gynecological oncologist led by Dr. Antonio Gil, who is the head of the department.